After confirming that with their protocol it was feasible to create 4-brain organoids, the authors then tried to use the same device, the same bioreactor, with a different approach in order to create organoids from the midbrain region. So they slightly modify the protocol that they use, changing the drugs added to the culture media in order to decide the fate of these iPSCs to midbrain. So after 18 days, they started to observe this TH marker, which is a marker of the midbrain cells. They, after the complete maturation of the organoids, they also observe this PITX3 marker and the TH marker. So all these markers in combination demonstrates that by changing the composition of the culture media, it is possible to obtain midbrain organoids instead of 4-brain organoids. So just to compare, they research for PAC6 and RAX1 markers, which are markers for 4-brain structures, and they see almost non-expression of these markers in these organoids. So we can say that this protocol was successful for midbrain organoids. Also, they tried to create hypothalamic organoids, so from the 4-brain going to this direction. Again, they changed the composition of the culture media, and they observe a lot of markers such as this NKX 2.1 and 0.2, also these two. So again, we can observe these organoids. Sorry about that. These organoids, which express several layers with different types of cells that resemble different brain structures in development. So in summary, yes, they were able to also differentiate these organoids into hypothalamic cells. So far, we covered the introduction, the hypothesis, experimental design, and some results of this study. So now we're gonna see how the authors did to evaluate the Zika infection mechanism in developing brains. So as a proof of principle, they started with a very simple molecule, BPA or bisphenol A, which is known to interrupt or disrupt the development of the brain in the fetus. So they started by adding different concentrations of this BPA molecule to the organoids, and then they evaluate how the drug or how this compound affects the organoids. So they found that the PPA reduces MPC proliferation or neural pluripotent cells proliferation, and which contributes to decrease the relative VC thickness. So if we remember, the VC thickness is this layer of the organoids which contains a lot of stem cells. It's very rich in these stem cells, and the cells will migrate from here to the outermost layers. So if this layer is decreased in thickness, this means that there will be not that much proliferation as in the control groups. So as a proof of principle, this worked. So after this, the authors started to infect the organoids with the Zika virus. So here they infected organoids at day 14 of formation, and they they let the virus go for 10 days and 18 days here. So on the organoids infected with the virus, they observe high expression of Cas3 marker, which we know that is a marker for cell death, compared to the control group. This is the group infected with the Zika virus. This is the control group. So we can observe the percentage of expression is significantly higher in the organisms infected. Also, they did this for they evaluate proliferation. If we remember, pH3 is a marker for proliferation. So in the control group, we can observe that there are some cells in the center of the organoids that highly express this marker, but in the organoids infected with the Zika virus, we don't see that much of expression of the pH3 marker. So the quantification of these assays also show a significantly lower expression of pH3. So in summary, increased cell death and decreased proliferative cells are pretty much the signs of microcephaly. So from day 14, we can conclude that the Zika virus can be correlated with microcephaly. They also did this for day 28. So they infected later or more mature organoids, and they let the virus for 4 days and 14 days here. Again, they did EDU analysis, which is also a marker for proliferation. For the control group, there was a high expression of EDU. For the group infected with the Zika virus, the EDU or proliferation marker was significantly lower, which we can see in here. And also this was dependent on the concentration of the Zika virus. So the higher the concentration, the less proliferation of the cells. We can also see in here how the Zika virus correlates more, or it's more likely to attack cells that also express SOX2, which we know that is a stem cell marker. So this stem cell marker, these two are markers for committed neurons. So we can see that there is a significantly higher infection of cells expressing stemness than other types of cells. So we can say that Zika virus attacks stem cells instead other types of cells, and this can explain why on adults, for example, Zika virus can be asymptomatic, because the pool of stem cells is not as high as in embryonic tissue. Lastly, for organoids of day 28, they also evaluate the thickness in the neuronal layer and the thickness in the BC layer. So we can see that in the control group, again, this is a highly organized organoid with a very thick BC layer with a high expression of SOX2. For the organoid infected with the Zika virus, we see that there is not that much of organization. It's hard to separate different layers in here. So also the thickness of the BC layer, which it's this pretty much, was significantly lower and was also dependent of concentration of the virus. So they repeated this for organoids on day 80. So they try to evaluate if the Zika virus infects other types of cells besides stem cells. They again observe that the infected cells correlate with the cells that are highly expressed SOX2 instead of those who express other type of committed neuron marker. We can see here the blue, which is a committed neuron marker. We don't see that much of the virus. So this is pretty much likely indicating that Zika virus is well correlated. The presence of Zika virus in the microenvironment of the cortical developing tissue can be correlated with signs of microcephaly. So let's move on to some limitations of this model. The first one is the high risk of necrotic course. This is because the higher the organoid is, it will be more difficult for the nutrients and oxygen to get through the innermost layers. So if this organoid becomes larger and larger, the cells in the center will be depleted of nutrients and oxygen and most likely will die. So this is why this model cannot replicate the human brain development beyond the second trimester of pregnancy. Also, even when some layers or there is some stratification in the organoids, also there is no representation of other zones or other layers in the normal cortical tissue. So it is known that, for example, this intermediate zone and subplay may play an important role in neuronal migration. So the lack of the representation of this tissue can be this model a little bit simplistic. Lastly, even when the cortical layers are separated to a certain point, they are not resemble exactly the structure of the forebrain tissue. So therefore the migration and positioning is not as in the tissue, in the normal tissue. So some future direction of this assay will be to use this same model to evaluate genes for schizophrenia or autistic spectrum disorders, because the authors show that there is some overlap in the upregulated and downregulated genes in the organoids that correlate to schizophrenia and autism. So this leads us to my proposed experiment. So it is known that the autism brain is different from normal brain, and it is suggested that there is an increased activity or number of connections in the neurons of the cerebral cortex.